Fantastic. We've got a really exciting session today. So with that, I'm going to introduce our first speaker, Rod Taylor, will be speaking to us about the rangeomorph Fractofusus. Are you there, Rod? Hello.
Okay, well, hello. Thank you, everybody, for coming along today. As Frankie said, I'm going to talk to you today about the Fractofusus misrai organism from the Ediacaran. This organism is found in rocks at Mistaken Point. And for those of you who don't know the area, probably not very many, it's located at the Mistaken Point Ecological Reserve in southeastern Newfoundland in eastern Canada. This area is protected by provincial regulations and legislation, so collection of fossils in this area is fortunately prohibited. There are actually several fossiliferous localities found at Mistaken Point. The main fossiliferous locality I want to talk about today has recently been dated to 565 million years ago. There are two large surfaces exposed in this area, generally referred to as the D and the E surfaces. They both represent deep water ocean floor environments that were rapidly buried by sediment-laden turbidity currents. And the most common organism on both of these surfaces is Fractofusus misrai. Here we can see a photograph of the E surface showing several specimens of Fractofusus, a large one across the bottom there and a few to the right, and a very small one towards the top left. We can also see here in the upper right and the lower left corners a grayish black material overlying part of the surface. This is actually the overlying tooth that was partly responsible for the preservation of the material at Mistaken Point. Fractofusus misrai was originally described in 2007 by Gehling and Narbonne. They described it as an elongate organism made up of a pair of parallel veins, which were connected to each other by a straight to weakly zigzag commissure or midline. Each of the two veins is made up of 15 or more separate modules, and each of these modules is in turn made up of one or more fractally branching frontlets. We can see here a reconstruction showing the complete organism and a magnification of one of those individual modules. Now, these modules are related to each other across the midline by a glide plane symmetry, so they're not actually symmetrical across the midline. Their conclusion in their manuscript was that the opposite surfaces of each of the two veins were apparently identical. In other words, they suggested that the top and the bottom surfaces of the organism were the same. And again, we can see here to the left their original reconstruction, and to the right we see a much more modern reconstruction produced just a couple of years ago by Charlotte Kenchington, looking at Fractofusus misrai from the top, and again reflecting this perspective that the top of the organism was the same as the bottom. If we think about the lifestyle of Fractofusus misrai, we know that it was an epifaunal recliner, which tells us basically that it grew very close or directly adpressed into the surface of the Ediacaran seafloor sediments. The distal tips of specimens, like the one we can see here, tend to be less well preserved, so it's quite possible that they were less well adhered to the seafloor compared to the main part of the organism. The branching of Fractofusus misrai is actually quite similar to what was originally described by Gehling and Narbonne. The organism is made up of two veins, which we refer to as branch rows, essentially using a more conventional rangeomorph terminology. And each of these two branch rows is made up of a series of individual primary order rangeomorph branches, which are tightly packed side to side. Now, as we can see here, the primary branches can be symmetrical across the midline. For example, across from just to the right of the green colored primary branch, we can see that the two individual primary branches are symmetrical. But if you go to the left of that green color branch, you can see that the symmetry across the midline very rapidly falls apart. So we definitely have glide plane symmetry in this organism. If we look in detail at one of these primary branches, we can see very easily the detail of the secondary branching. There is a medial secondary branch coming directly from the midline that bifurcates multiple times and widens distally as you get towards the margin of the organism. Sorry, I'm having issues with sunlight here and shading. If we look to the left and the right of this medial branch, we can see what we refer to as subsidiary secondary branches. These are found on both sides of the primary branch and are non-branching. If we look slightly more closely, we can see that tertiary branching is found as well. In fact, what we've done here is I've colored in two of the subsidiary branches just to show a comparison. The subsidiary branch on the left, we can see on both the left and the right margins, there are very clear traces of the tertiary branching visible. In the right secondary branch, however, we can only see the tertiary branches visible on the left edge of that secondary branch. I'm going to come back to this later because this is an important taphonomic feature. One of the things we find in looking at the specimens at mistaken point of Fractofusus misrai is that there's considerable variation in the overall morphology that we see. Many of the specimens, as we can see here, are preserved as very simple straight lines, and we can actually see here 3 separate specimens that show the straight orientation quite clearly. Another relatively common form that we see are what we refer to as curved specimens. And we can see along the bottom of the slide here, a nice long specimen with a nice gentle S-shaped curve. And to the upper left, a smaller specimen showing a similar curvature. Here we see a slightly, a considerably less common form that we refer to as kinked. And we can see that approximately 2/3 of the body of this organism is in a straight line, almost parallel to the photograph. But as you get towards the left edge of the specimen, we can see there's a dramatic change in direction where we have a specific point at which the direction of the organism changes, and the rest moves in more than a 90-degree direction away from the remainder of the specimen. And we even find some specimens that possess multiple variations in their shape. So this is a specimen that shows considerably complicated curvature along the length of the organism. So these variations in shape are, as I say, there's a lot of different variations and forms that we see. They're probably due to actually changes in direction of growth in the organism itself, which was probably caused by response to environmental gradients. For example, it may have been in response to nutrients or toxins in the environment around them. It could have been in response to physical proximity or contact with other specimens, or it could have even been related to mechanical stimulation caused by water currents in the environment around them. It's worth pointing out here that changes in direction of growth for an organism like Fractofusus misrai would actually have been relatively easy to occur if the addition of new terminal branches was the main factor associated with growth, because all you need to do is stick in a larger or an extra branch at one of those 2 branch rows, and you'll automatically develop a curve in there. Now, it's worth pointing out that some of these curves and kinks may also be caused by the organism being susceptible to lift in the environment, caused by water flow in the area around them. So portions of the edges may have been unequally exposed to sediment-laden currents, so parts of the organism may have actually been more easy to lift or to move about than other parts. In order to try to understand these specimens better and the variations that we see, we looked at as many specimens as we could for which we could get a reliable count of primary branches along both primary branch rows, and we found a total of 75 specimens for which we could get reliable counts. Of these, we found that 52 specimens had at least one additional primary branch on the convex surface of the specimen. So there does definitely appear to be a correlation between the shape of the organisms and the addition of new branches. One of the factors that we see associated with curvature and kinks is branch disruption. We tend to see 2 different types of branch disruption on these organisms, and we refer to them as branch compression and branch overlap. And what these compression features tell us is that these individual branches appear to be fixed to the organism at the midline, but they probably weren't actually connected to each other laterally. I will show you a couple of examples of these, and hopefully you'll agree with me on this. We can see here a specimen with a very distinct curvature to the right. And on the inside margin of that curve, I've colored in 2 primary branches. And we can actually see here how, as we go from the medial portion of the organism to the outside, the lateral margin, we actually have distinct narrowing as these branches have basically been pressed together because of the change in direction of growth. Here we see a specimen that's commonly referred to in the literature as a folded specimen. We've colored in the 2 portions of the organism in different colors. So the larger portion is an orangey color and the smaller portion is in yellow. And we can quite clearly see that one of these overlays the other considerably. Now, I will point out very quickly here that we've even colored in an extra section to the far left, very close to that folded region, a single pink branch. And we've colored this one in because in the rest of the organism, the yellow and the orange sections, we can see, if we look at this closely, most of the secondary branches are aligned relatively closely to the positions we'd expect them to be in. In the pink branch, the actual secondary branches are considerably moved about and changed in position. Now, I've got another specimen here that shows this more effectively. It's rather heavily eroded, as we can see, especially towards the right, but I've colored in some of the primary branches on the left, and we can actually see, I hope quite easily, that there's a lot of variation in the size, the shape, the position and the orientation of these primary branches. They definitely aren't lying in that tightly knit position that we tend to see in most specimens. And what we think is happening here is that these branches have actually been tousled. So the specimen has been disrupted somehow, perhaps by currents in the water around them, causing these primary branches to lift slightly off the sediment surface and actually redeposit again after the environmental disturbances passed, causing them to settle in a position considerably different than they had been when they were originally growing. And these disruptions can actually be quite disturbing for these organisms, as we can see here. This is a specimen with a very dramatic kink, almost 90 degrees change in the direction of the organism's growth. And on the convex surface of that actual kink, we can see where the bright white arrow is. There's a big chunk of the organism actually missing or torn away. And on the far left side of the organism, where we would have expected to see a nice tapering distal end, we actually have a rather abrupt, clean break on the organism. So it appears that some of these forms of movement that would have caused these specimens to kink or to curve could actually have been quite damaging to the organisms. Another important factor that we see with these organisms that varies considerably is the actual outer margins that we can see. And here we've got an example of what we call a linear margin. And I hope you can see here where we've colored them with blue. The outside margin of the specimen is not quite in a straight line, but it's actually a continuous line across several primary branches. So even though these branches were independent, at times it looks like the outer margin of the organism is continuous across the outside surface of the specimen. If we compare that to other specimens, such as this one here, I've colored in the distal tips of one single primary branch, and I hope you can see what we refer to here as a scalloped margin, where you've got a very clear curved margin for each of the secondary branches in that primary branch. So there's considerable change between different specimens that we can see with the actual lateral margins. Excuse me. What this edge morphology tells us, these variations, is that the distal tips of these organisms could probably have been lifted from the seafloor, more than likely by currents in the area around them. Lifting the edges of these branches would have allowed sediment to accumulate underneath the organism, which would have erased the impression that the specimen would have made lying on the sediment surface and would have produced an irregular but relatively linear margin, as we saw in the first example. The scallop margins that we see, we believe, developed when these organisms remained in contact with the seafloor and were actually buried with these specimens in position, so the details of the curvature of the primary margins would have been preserved. Now, I referred earlier to a folded specimen, and these have actually been used in the past to suggest that the upper and lower surfaces of Fractofusus misrai were the same. As with the folded specimens like the one I showed you earlier, both of the two sections of the specimen appear to show similar morphological features. However, if we consider the morphological model that we're presenting here, if you take a specimen that has loose independent branching or branches that are attached to the midline, pick it up and flip it over, it's incredibly unlikely that those independent primary branches are going to settle down again in the same position and orientation that they had been in their original growth position. It's considerably more likely that these folded specimens actually represent an exaggerated form of what we call a kinked specimen. So at the point of the kink, instead of just having a part of the specimen rotate sideways, it actually picks up and rotates and partially overlaps the original portion or the larger portion of the specimen. So we're seeing an overlap here related to a rotation instead of a fold. And in this case, both of the two parts of the specimen that are still in contact with the surface have their lower surface resting on the sediment surface, which explains the similarity that we see in the branching there. Sorry, wrong button. And here I'd like to show you a reconstruction of Fractofusus misrai, looking at it from the upper surface. And we can see here again, we've got two veins or primary branch rows made up of a series of primary branches. We can see that there's a potential midline. This is a hypothetical structure because we don't actually have specimens preserved with the upper surface preserved, but there must have been some kind of a medial structure for these primary branches to attach to. But we can see that the secondary branches within each of the primary branches are independent, they're loose, and they were closely packed together, making each of these primary branches fairly tightly compressed together. And on the lower part, we can see it was probably a fairly low-lying flat organism on the seafloor. So to conclude, Fractofusus misrai was a complex rangeomorph with distinctly different and upper-lower surfaces. The distal branch tips rose upwards from the seafloor and could probably move about under seafloor currents. We think that it utilized chemosymbiosis for feeding because it could probably bring fresh water underneath the surface, leading to oxygenation and bacterial growth. And we realize here, this study tells us that you really need to understand the taphonomy of these organisms before you can understand the morphology and the biology of these organisms. And I'm very close to out of time, so I will say thank you and let you read my acknowledgements while I answer any questions that may be out there. Thank you very much.
Very much for that talk, Rod. That was fantastic. I quite enjoyed Fractofusus. Particularly in
Sorry, I went too long. My apologies.
No, I'm afraid we don't have time for questions. We're on quite a tight timetable, but there are questions for you waiting in the chat. So maybe during the next talk you could answer them. There are quite a few in fact.
Thank you.
And with that, I'm going to introduce our next speaker, Catherine Boddy. Hello, Kat.
Hi.
I'm just going to start your presentation now.
All right. Hi, Rachel. How are you?
Awesome. Thank you very much, and thanks everyone for coming to my talk. I'm going to be talking to you about the macrobiota and how they were distributed across the globe during the Ediacaran, and whether the palaeogeography influenced their distribution. So the Ediacaran period is an important area of study as it marks the transition from microscopic eukaryotes to the radiation of macroscopic life. And the diversity of Ediacaran macrobiota can provide insight into the patterns and processes behind the diversification of complex life. And whilst previous work has looked at how the effects of age, paleoenvironment, and lithology on Ediacaran diversity, here we're going to be looking at the effects of paleogeography. So the Ediacaran looks a little bit different to how it does today. On the modern map at the top, I've shaded the Precambrian crust, and on the lower map, I've highlighted where this crust is thought to have been during the Ediacaran. However, Precambrian paleogeographic reconstructions are highly debated, especially with the positioning of Laurentia, Siberia, and Baltica. So we compiled a comprehensive database from the literature, including of macroscopic taxa and where they're found across 31 localities around the world. There are two main models, modeling techniques behind global plate reconstructions. So we plotted our data on one of each. And these models are continental drift models and full plate tectonic models, and the latter includes more data, so we think they're more accurate, and therefore therefore this talk will show our data on that reconstruction. We also considered the environmental and lithological controls by plotting them on maps shown at the bottom to see whether they were affecting our paleogeographic signals. And finally, we performed statistical tests to analyze whether our results were significant or not. So here I show you the overall diversity of Ediacaran macrobiota across 10 million year time intervals. I've got pie charts on each locality scaled to the number of taxa at each place. And you can see from the start in the top left map, despite there being a sparse distribution of localities, you can see that protists in the bright blue sections and frondose taxa in the pale blue sections of the pie chart were globally distributed across both high and low latitudes. And such a global distribution may be a result of a longer history prior to what we observe in the late Ediacaran, or perhaps the inclusion of a waterborne rapid waterborne dispersal phase in their reproduction, allowing them to radiate across the globe rapidly. Another thing that I'd like to point out to you on these maps are especially visible in the later two time bins at the bottom of the screen. Their diversity appears to be greater at low latitudes. So that leads us to think, was there a latitudinal biodiversity gradient during the late Ediacaran? In the modern day, biodiversity follows a general trend where there's higher diversity at lower latitudes, which decreases towards the poles. This pattern has been observed at various points throughout history with varying magnitude and gradient. And this is thought to be due to the dominant climate regime of the time. So during the Ediacaran, if I plot the raw taxonomic density across latitudes, it appears that there's higher diversity at low latitudes, which decreases towards the poles. However, when I then impose the number of localities that we have across the latitudes, you can see that this gradient roughly tracks the sampling bias that we have on these from this time. And if I took this blue line away from the taxonomic diversity in the orange, represented by the orange line, to see the deviations from the expected distribution if sampling bias was uniform and if the expected distribution were uniform across all latitudes, you can see that the deviations are minimal and we can't actually distinguish latitudinal biodiversity gradient from the sampling bias during the Ediacaran. So going back to our diversity through time maps, another thing that I'd like to point out to you is the apparent increase in diversity between the second time bin, which is shown in the top right map, and the third time bin shown in the bottom left map. And finally, and subsequently, a decrease in diversity in the final time bin in the bottom right. These two patterns have been suggested to be representing a second wave of ecological innovation between the second and third time bin, and the diversity dip towards the end has been suggested to be due to an extinction event. However, again, when we impose the distribution of localities through time, the pattern roughly tracks this, and we can't actually statistically distinguish neither of these patterns in our data. So if we now look at individual groups following this trend through time to see how they were behaving, and to see how paleogeography had a role, I've split them into two 45 degree latitudinal bands here. And now we can see how paleolatitude was affecting individual groups. The first one that I'd like to point out to you are these bilateral taxa, which I've now highlighted with a red outline. And you can see that they radiate later than a few of the other groups during the Ediacaran, and they're also only found at low latitudes. So these bilateral taxa include taxa with bilateral symmetry. These include Cambrian, which I've shown an example of on the left image, Dickinsonian, which I've shown an image of in the right image, and bilateralomorpha. These three groups include some of the most likely candidate stem bilaterian taxa. So looking at where these are distributed across the globe, we can see that all body fossils from each of these three groups are found entirely within the lowest 30-degree latitudinal band, with a few trace fossils that are thought to be produced by bilateral taxa at high latitudes towards the very end of the late Ediacaran. So plotting these across latitude, you can see that the taxa now deviates quite significantly away from the blue localities line, and above the line at low latitudes and below the line at high latitudes. And if I again take that blue localities line away from the taxon line, you can see that the deviation away from the expected distribution, if it were uniform, and if sampling was uniform across all latitudes, you can see that there's a significant difference between an increased taxonomic diversity at low latitudes and lower taxonomic diversity at high latitudes. So what this pattern might mean is that perhaps bilateral taxa evolved later, or perhaps more slowly than other taxon types during the Ediacaran, found during the Ediacaran. This is consistent with phylogenetic expectations that bilaterian taxa developed after non-bilaterian metazoans. And perhaps because they're only found at low latitudes, perhaps they had a temperature dependence, preferring the higher temperatures that are found across the equator, and that these low latitudes may have provided a cradle of preferential conditions for bilaterian evolution. So going back to our split latitude charts of all the taxa and all the groups through time. Another group that I'd like to point out to you are the tubular taxa, which I've now highlighted with a red outline. So as with the bilateral taxa, we can see that they developed later in the Ediacaran than other groups, but this time they're found at both low and high latitudes. So along with the appearance of tubular taxa, we also had the appearance of hard biomineralizing skeletons, which also developed towards the end of the Ediacaran. Most of these biomineralizing taxa are tubular during the Ediacaran. And if I show you the distribution of them across the globe, with the solid pie chart showing the biomineralizing taxa, with pink sections of these pie charts being the tubular biomineralizing taxa, and then the faint circles showing all tubular taxa, we can see that both tubular taxa and biomineralizing taxa, including the biomineralizing tubular taxa, are found globally distributed across the, with no obvious difference between low and high latitudes. So in contrast to the bilateral taxa that developed later but were only found at low latitudes, this may suggest that they had, the global distribution of biomineralizing and tubular taxa may suggest that they had a rapid global radiation. And the process of biomineralization is thought to have, prefers warmer water conditions. And so it has been thought that these taxa would be found preferentially at lower paleolatitudes. However, our data suggests that they were everywhere, suggesting that, which opens up new avenues for further work. Perhaps they were found at higher latitudes because the global temperature was warmer, providing warm water conditions at high latitudes as well as low latitudes. Or perhaps if the ocean chemistry were more preferential for biomineralization, then biomineralization could have also occurred in cool water temperatures at high latitudes. Alternatively, perhaps these biomineralizing taxa weren't actually biomineralizing at all, and the hard parts that we see are all formed diagenetically. So, in conclusion, paleolatitude does appear to have exerted a significant control on the distribution of some taxonomic groups during the Ediacaran, most obviously the bilateral taxa. So therefore, paleolatitude should be considered alongside other factors that are known to have affected the distribution and diversity of Ediacaran macrobiota. Finally, taxonomic diversity patterns through time cannot be distinguished from sampling bias in our data, including a diversity dip, which may suggest an extinction event at the end of the Ediacaran. So thank you very much for listening. Thank you, Palace, for putting on a great conference. And thank you to all the people and organisations on the screen for all of the support.
Thank you, Kat. That was an absolutely wonderful talk. Loads of really interesting things to be made. And we have plenty of time for questions. I'm sure you'll be thrilled to we already have a couple coming in. So one from Lucy Muir. I'm intrigued by the high number of sponges in the assemblages. What are you classifying as a sponge?
So these are, we note that we don't actually, given that sponges do have quite an obvious phylogenetic interpretation, we don't actually know whether these were sponges, but we've included taxa that have pores that have been suggested to be for filter feeding, although we can't see any spicules in them. But there are only a few taxa that we've included in this group.
Another one coming from Ross Anderson. Given these deposits are instances of exceptional preservation, might we also expect taphonomic processes to vary by latitude?
This is a good question. I think obviously the production of carbonates is thought to be different with temperature, but again, if we're seeing them everywhere, then that changes. I'm not sure. I'll have to think about that a bit more. But
Lots of avenues for future research.
Yeah.
One's come in from Joe Flannery Sutherland. How does assemblage composition vary with depositional environment? Might that affect evidence of an LDG given the distribution of depositional environments?
So we find that the depositional environments are found at both low and high latitudes. So we don't think that they're particularly affecting the gradient there. We performed a hierarchical cluster analysis, and we found that assemblages were separating out, but they were not particularly separated out by the depositional environment, apart from in a few smaller subgroups. So I wouldn't say that depositional environment is particularly affecting it.
We have time for one more very quick question from Duncan, and then there are a couple more in the chat, but we have to move on, so I'll just ask you to answer those ones in the chat. So Dima asks, how did you solve the problem with Ediacaran taxonomy that has been littered with junior synonyms, pseudotaxa, different taphonomic variants, and ontogenetic stages having separate taxonomic names?
So we looked at each individual one separately, and we decided we split everything by just genera rather than species names, so that we avoided oversplitting of certain genera, and we considered the literature as up to date as we possibly could to use those interpretations and keep it up to date.
Fantastic. Thank you so much, Kat. It was a really interesting talk. As I said, there are just a few more questions for you in the chat. And with that, I think we'll move on to our next speaker, who is Ben Slater. Are you there, Ben?
Yep, here.
Hello. Great.
Hey.
I'll just start your slideshow for you.
Thanks. Okay, thanks a lot, Frankie, and hey, everyone. So the Ediacaran-Cambrian boundary sits close to the most dramatic ecological and evolutionary transition in Earth history. And it's somewhere around this point in time that the fossil record itself changes forever. So, and most and sedimentary rocks younger than this boundary contain most of the fossil record as we know it. So, although all kind of Cambrian taxa and indeed all living organisms must be descended from lineages that survived from the Ediacaran period, the impression that we get from the current fossil record is instead one of a fairly sort of stark disconnect or discontinuity between Ediacaran and Cambrian biotas. So, for example, if you look at a Lagerstätten from the late Ediacaran and compare it to one from the early Cambrian, there are more or less no shared taxa. And in fact, very few body fossil taxa at all are known to span this boundary. So there are some kind of rare cases. For example, the macrofossils Swartpuntia and Pteridinium potentially range just into the base of the Cambrian system in sections in Namibia. And there's also a limited variety of more or less kind of broadly tubular fossils that were once thought to be restricted to either late Ediacaran rocks in the case of Claudina, for example, or to Cambrian Fortunian strata, like these various kind of organic tubes that you can see on the right here. But recent scrutiny of the fossil records of these groups has revealed that they actually range across the Ediacaran-Cambrian boundary. And we can also add to this list of so-called, we can also add to this list of so-called Ediacaran survivors a small but increasing number of taxa that purely on morphological grounds have been linked to the Ediacaran biota. And these include things like the Cambrian fossil Stromataveris, which was recently redescribed by Jennifer Hoyle-Cuthill and Jan Han as comparable to various Ediacaran Frondos taxa, and also Thalmaptylan, wrong direction, and also Thalmaptylan from the Burgess Shale, which has been compared to Ediacaran taxa like Charnia discus. So these taxa themselves don't strictly range across the boundary, but if we're generous, we can add them to the list of kind of Ediacaran survivors. They look like kind of hangovers from the Ediacaran biota. So despite these kind of rare exceptions, whichever way you look at it, there's still only a fairly kind of modest number of body fossil taxa that demonstrably span this boundary. So there are lineages for which we have Proterozoic fossil records that must sail across this boundary, like various kind of prokaryote clades and crown group eukaryotes like red algae, green algae, and protists like testate amoebae. But it's still fair to say that there are very few macrofossils of specific taxa that cross from the Ediacaran into the Cambrian. And this apparent disconnect has led to suggestions that Phanerozoic itself may have in fact begun with a kind of a mass extinction of sorts, perhaps eliminating the familiar Ediacaran biota, followed by a kind of wholesale replacement by the classical Cambrian fauna. And although this would echo patterns of extinction and radiation that we see later in the Phanerozoic, it's been pointed out that the case for extinction here is not so straightforward, since the fossil record around this transition sees major changes in patterns of fossil preservation itself, with several kind of major taphonomic windows opening and closing at this time. You might have seen Dima Duncan's talk this morning about this topic. So, for example, much of our view of biotas from this time period comes from Lagerstätten sites, so Burgess shale type sites in the Cambrian, which preserve carbonaceous compressions of soft-bodied fauna. And then in the late Ediacaran, these sites preserving cast and mold fossils of soft-bodied organisms in so-called Ediacara type preservation. Unfortunately, though, there's actually quite a substantial and significant temporal gap between these preservation types right around this transition. So Ediacara style preservation, for example, seems to disappear around the Cambrian boundary around 540 million years ago or so, whereas all of the Cambrian Burgess shale type sites don't appear until well into the Cambrian. And this pattern has kind of left this sort of missing window of almost 20 to 30 million years or so sitting right around the boundary, and particularly in the earliest Cambrian, where we have very little data on soft-bodied forms of life, which is, of course, the majority of diversity. And to make matters worse, there's not just a temporal gap, but there also seems to be pretty kind of little taphonomic overlap between these different styles of preservation. So only a few taxa have actually been found fossilized in both these types of Lagerstätten. There's Flabellophyton, a macroalga, and also Eoandromeda, this possible stem group ctenophore that you can see here is perhaps the best known example. But in general, the lack of overlap between these different taphonomic modes could also be kind of amplifying the apparent disconnect. But unlike the Lagerstätten records, the more kind of unexceptional records of shelly fossils and trace fossils likely represent more continuous records through this interval. But since the record of shelly fossils essentially begins around this time, they can tell us a lot about originations occurring in the early Cambrian, but they're kind of little use in tracking Ediacaran clades, since there's only a handful of Ediacaran biomineralizers to begin with. Likewise, trace fossils show a pattern of continuity from the late Ediacaran into the early Cambrian. So they're kind of an excellent means of examining this transition, but they only record benthic bilaterian behavior. So ideally what we need is a more kind of continuous record of soft-bodied fossils covering, soft-bodied body fossils covering this interval. And one of the ways we might start building up such a record is by exploiting a property of Burgess shale type preservation itself. So because fossils like this Wawaxia here are preserved as flattened carbonaceous compressions, it's possible to actually dissolve away the rock matrix using hydrofluoric acid and extract and sieve out the isolated kind of organic debris and elements that are left behind. And this is interesting because it demonstrates that fragments like these might be preserved as dispersed elements outside the Lagerstätten sites, where you don't get these kind of special conditions required to form the fully articulated macrofossils. And it's turning out that there is this rich record of unmineralized metazoans occurring as disarticulated fragments. So you might have seen Elise Wallett's talk yesterday about some of the latest Cambrian SCF finds that are coming out. And these have been dubbed small carbonaceous fossils, or SCFs, because like small shelly fossils, they're essentially just a polyphyletic sort of mixture of small animal fragments. So by using these techniques and looking at these fossils, these SCFs, recently it's been possible to really expand the spatial and temporal records of groups like Wawaxids, hallucinogenic lobopods, Cambrian annelids, priapulids, all groups that are otherwise pretty much confined to Lagerstätten. So through tracking these kind of distinctive fragments, we can really improve our records of soft-bodied animal groups in the Cambrian. And significantly, perhaps most significantly, we've recently been able to kind of extend this SCF's record right into the earliest Cambrian Fortunian, where there are no Burgess shale type Lagerstätten to speak of. So the next kind of question is it possible to push this record further back and track these kinds of fossils across the Ediacaran Cambrian boundary, where we have this seeming disconnect in other modes of preservation, but where SCFs likely have a much more continuous record? So recently we've been examining earliest Cambrian and latest Ediacaran deposits for SCFs. And one particularly interesting SCF that's caught our attention are these distinctive spiral forms known as Cochleatina. And what we did is we examined sediments from the late Ediacaran of the Baltic region and found that these Cochleatina were fairly abundant in mudstones from this age. And so these examples here are from the late Ediacaran of Estonia, and you can see that they're often fused to these kind of organic sheets and even occur as clusters, so seemingly in sort of triplets of 3 identical coils per cluster. And these things appear to be abundant in late Ediacaran deposits in multiple different localities. So we also recovered Cochleatina from the late Ediacaran Krushanovka formation of Ukraine. So both Estonia and Ukraine were both part of this paleocontinental bloc Baltica during this time. But there were actually some sporadic reports of Cochleatina from other paleocontinents as well. So especially as those that have been recovered as fragments among palaeontological preparations. Sometimes these have been identified as Cochleatina, and sometimes they're kind of been misassigned to certain sort of coiled filamentous taxa. But when we synthesized all these isolated reports, different isolated reports together, you can see that the vast majority of occurrences have been documented from the East European platform on Baltica. You can see up in the left here. But there are also scattered reports from Siberia and from Perigondwanan terrains, showing that Cochleatina was likely sort of globally or at least very widely distributed in the late Ediacaran. And further, when we combo all these isolated reports together and plot them stratigraphically, a very distinct pattern emerges. And it's clear that Cochleatina ranges well across the Ediacaran Cambrian boundary. And in fact, when you compare it to other sorts of Ediacaran survivors, it's one of the only taxa of these that with a complex morphology that actually spans a substantial portion of the late Ediacaran and also ranges well into the Fortunian. So another kind of obvious next question is, what is Cochleatina? What kind of organism does it belong to? So like many Ediacaran fossils, the precise biological affinities of Cochleatina are unknown, and it's previously been compared to all sorts of organisms from right across the tree of life, including things like the coiled radulae of mollusks, other Ediacaran fossils like Redkenia, and even the coiled elaters of plant spores in some kind of more unusual comparisons. So whilst there are some pretty interesting potential functional analogues with a few of these structures, when we looked at the microstructure of Cochleatina in detail, none of these kind of comparisons really stands up to scrutiny. So, for example, the broad similarity to the overall sort of shape of mollusk radulae can probably be discounted because Cochleatina itself is actually formed as a kind of solid ribbon-like structure rather than a series of tooth-like elements, as you would see in a kind of mollusk radula. And also the triplets and the clusters don't make much sense as a radula-type mouthpart. So while we still can't say for sure what kind of clade Cochleatina belongs to, we've at least been able to kind of rule out some of the past sort of previous interpretations. And so to sum up some of the findings here, Cochleatina stands as a really nice case study, sort of highlighting the potential of SCFs as a really novel way of tracking evolutionary patterns across the Proterozoic-Phanerozoic transition. And crucially, Cochleatina shows that SCFs can capture unmineralized taxa, but unlike Lagerstätten, this type of preservation doesn't really seem to have a restricted taphonomic window, at least not in time. So it can provide a relatively sort of continuous record from a really broad range of paleo environments. So it's great for looking at this transition. So thanks very much for listening, and I'd be happy to take any questions.
Very much for that talk, Ben. That was really interesting. We have got questions coming in and plenty of time for them. So Ross Anderson asks, why do you think we have deposits with SCF in the lower Cambrian but no deposits with macroscopic BST fossils? How similar are these modes of preservation? And secondly, are there paleogeographic biases to the SCF record in general?
That's a great question, yeah. So I'm not sure why there's no kind of macroscopic Burgess shell type deposits in, say, the Fortunian. There are some kind of like occasional like tubes and macroalgae that are preserved as like bedding plane compressions, but there doesn't seem to be anything on the scale of like the Berdych Shale or the Chenjiang. And I think that's something to do with, it must be something to do with the formation of these abrution deposits or some kind of chance conditions that makes it Berdych Shale type, macroscopic Berdych Shale type preservation more common later in the early Cambrian. Whereas SCFs, although they're kind of carbonaceous compressions, so they're the same as Berdych Shale type macrofossils in that respect, they don't have the kind of biostratonomic limitations. So perhaps it's just easy. They're more like aquitacs in that respect, and they could probably preserve in a much more wide range of depositional environments. In terms of biogeography, I'm not sure about if there's any kind of obvious biogeographic signal among SCFs yet. Like, we still need to look at more deposits, I think. But I can't see any reason why there would be a kind of major biogeographic trend or bias, anyway. Oh, sorry, I can't hear you, Frankie.
Sorry, I
It's all right.
turned my microphone on. We'll have two more questions, which we'll just rattle through quite quickly. One from Jeff Thompson. Hi, do you think these SCFs have utility for constraining divergences in molecular clock type analyses? Do you have any reliable crown group features for some clades?
Yeah, of course. I mean, like any fossils, I think they'd be useful for constraining molecular clocks. But so in the Cambrian, there are a few kind of fossils of potential crown group crustaceans. If you saw Elise Wallett's talk, and also work by Tom Harvey, has shown lots of these kind of mandibles and filter feeding mouthparts that we don't see elsewhere in Cambrian deposits. We don't see them in like Berdych Shale type Lagerstätten, for example. And they're potentially crown group crustaceans that are much earlier than the next kind of earliest crown group representatives in the fossil record. And they're obviously really useful for calibrating molecular clocks. In the Ediacaran, nothing so far that can be, there's some potential crown group, potential total group bilaterians starting to turn up, but it's a bit, no kind of crown group representatives of specific clades so far.
Fantastic. I've just looked at the time. I don't think we have any more questions, but there are two really interesting ones in the chat from Luke Parry and Christopher Whalen. If you could answer them in the next talk, maybe, that would be fab.
Yeah, sure.
Otherwise, just say thank you, Ben. That was fantastic. And we'll move on to our next speaker.
Thanks a lot.
Javier Ortega Hernandez. Welcome, Javier. I'm just going to start your presentation now.
Okay, awesome. Thank you so much. It's great to be here. So let's get started. Before we go off, this presentation has some interactive elements to it. So if you have your phone at hand, as we all do in this day and age, I can recommend that you open your camera app, and then you will see two instances of these QR codes throughout the presentation. So if you scan it with your camera, you will be able to interact directly with some of the microcity models that we're going to talk about today. So I do hope you take this opportunity to enjoy them. So the actual talk itself is going to be about the panarthropod head problem. Now, the panarthropod head problem is the shorthand we use for talking about the complications involved in comparing between very phylogenetically distant groups of panarthropods. Of course, panarthropods are very disparate themselves, and this makes it that it can be quite hard to identify homologs between all of these different groups. Broadly speaking, all panarthropods have a head, right? And this head has fundamentally three segments at the very, in general. So this would be the protocerebrum, which I've highlighted in blue, the deuterocerebrum, which I have highlighted in red, and the tritocerebrum that is only really present in arthropods, but let's say for the sake of argument, that is the segment we will be talking about. So what we're seeing here is basically how we understand this serial homology among extant panarthropods. And that's great, but it has been the result of a lot of work, particularly between embryology, morphology, and development, as well as nervous system. Now, at the center of the panarthropod head problem, we have this structure called the labrum, which really breaks everyone's heads all the time because it is a really complicated thing. The labrum, for those of you that may not necessarily know top of your head what this is basically a flap-like structure that sits on top of the mouth opening. So In this case of this lovely giant isopod, it basically represents this clypeolabral complex, and it represents the roof of the mouth so that they can sort of masticate stuff. However, the labrum has received a lot of interest because it has some potential implications in linking extant arthropods with some of their Cambrian representatives. Also, the labrum has a very controversial history in that it may belong to a pair of ancestral limbs, and there have been a number of hypotheses trying to explain its origin and evolution. Now, of course, we want to look at this from a deep time perspective, and in this case, the Cambrian fossil record is both fantastic and absolutely horrible, because, of course, fossils do provide us with this ancestral morphology that we can then compare with extant diversity, and we can start to piece together the gradual evolution of all of these important features. The problem is that, of course, fossils being what they are often flat, and only until recently we have been able to gain a much better understanding of the segmentation of some of these organisms. Now, because of the complications associated with dealing with the arthropod fossil record, which includes both what we call lower stem arthropods, such as Achaea, Cricmachila, and Anomalcaris, so animals that do not have, for the most part, an arthrodized exoskeleton, we also have this so-called upper stem, which includes animals that we would not doubt to call, oh, they are arthropods. They have the jointed legs. They are clearly arthropods, even if not crown group. Now, the complication here is that basically every hypothesis under the sun has been suggested to try to account for what is the segmental homology between the appendages of all of these very different animals. What we can agree on is that the labrum, as we understand it from extant diversity, has a protocerebral origin. So the labrum does belong to the anterior-most part of the brain and the head. So that's good enough already. And extant arthropods have a labrum, and they also have a deuterocerebral first limb pair. So in mandibulates, this would be the antenule or the first antenna, and in chelicerates, it would be the chelicera. In all of those cases, they have a labrum. The problem is that when we see the fossils, it is very hard to identify how this serial homology works because, of course, we only have very little information from them, and that's even coming from the most exceptional fossils that we have at hand. And in particular, megachirons are a real pain because every single position for the great appendage derivation has been suggested at some point or another, including the protocerebrum, the deuterocerebrum, and the tritocerebrum. And depending where these great appendages come from, these very kind of iconic sort of raptorial limbs, we have very different implications for whether the labrum exists in megachirons, and if so, what appendage these great appendages actually, what segment these great appendages come from. So that is what we're going to explore today. So as we mentioned before, Cambrian fossils from Burgess shell type deposits, wonderful as they are kind of limited because they are quite flattened in many cases. And even though we have wonderful external morphological information, we often do not have a lot of internal data to go with. And the compression that these fossils experience can make it quite challenging to actually identify all of these different elements. So what can we do about it? So Frankie, if you can please bring the first video, we can show what we have been up to recently. And that is to employ micro-CT scanning to actually try to get more information from these fossils. This is showing just the core elements of how micro-CT scanning works, although I realize I may be preaching to the choir here. But basically, we can take advantage of the preservation of fossils in the Chengjiang deposit in South China because they are very highly paratized. Now, because of the prioritization, this produces an extremely strong contrast between the fossil, the body fossil itself, and the surrounding matrix, which is what we see here. So what you will see is this kind of white blob that appears, and this will repeat in a second. That is a fossil that we actually want to look at. And because we can kind of digitally dissect this out, we're able to actually observe the morphology in great detail. So for the purposes of this talk, we're going to check out the detailed morphology of the megacharon, the Linquelid lesabrosa from the Chengjiang. Again, there's a secure code. Please go ahead and do scan it with your camera app, and then do enjoy the 3D spinning model at the fingertips. So what is interesting about Nicholls lesabrosa is that this animal is extremely well known. There are at least half a dozen publications that deal with its morphology. And more importantly, it would be difficult to find something new to say about it because there are hundreds of specimens. Again, it is a very well understood animal. So what we do here is, of course, employ microcity work and try to get that little extra juicy detail to get additional information from this organism. So if we can see the next video, I'm going to show you one of the virtual models that we were able to obtain from this data. And this is a model of a 7 millimeter long juvenile of Nicholls lesabrosa. What you can see is that it just has so much lovely morphology inside of it. You can see the great appendages, you can see all the limbs on the body. I mean, it's really fantastic. The details of the endites, the podomeres, tail spine. This is going to spin around in a second. And what is really interesting is that we can also get some taphonomic information here. You may notice that in the middle of the specimen, there is a rectangular hole, like just looks black. And that is because that would be the position of the gut, or at least the foregut, that would be filled with sediment. So because the sediment has the same composition as the matrix, we don't really see it in the scan. But we do get to see all the other parts of the parotized anatomy. So we were able to do this with two specimens, one 7 millimeter larva or juvenile and one 5 millimeter juvenile. And we basically got really interesting information about the ventral organization of the head. So in this case, what I'm showing you on the left side of the screen in panel B is a magnification of the head of the 7 millimeter larva with the great appendages removed. So we can see the head in much greater detail. Again, if you want to see the same model in 3D, please go ahead and scan the QR code and enjoy. And what is really significant here, and what really kind of made my jaw drop when I saw this for the first time, is that once we remove the great appendages, we get a really clear view of the mouth opening. So what we have here is, of course, the head shield, which is this kind of semicircular carapace. But posterior to that, we have four eyes or 2 eye doublets. And behind that, we have this structure that we have labeled as LA, which would be the labrum itself. Now, we know that this is a labrum because it has a very peculiar morphology. It is a single structure. It is quite clearly convex. And critically, this is directly associated with the mouth opening. As we mentioned before, we can see the preservation of the foregut, and that basically transitions into the mouth. The mouth here is indicated by the arrow, and as you can see, there's just a hole in there. And that is where all the sediment would basically come in, and that is the reason why it doesn't get picked up by the CT scan. In panel C, we can see this on a sagittal section, and what this is showing us is basically the first part of the alimentary canal going into the body itself, even though this has been quite compressed. Now, for comparison, it's really interesting that there are very close analogs, or homologs maybe, with what we see in the embryology of extant arthropods. In panel D, we have an embryo of a spider where it shows the labrum. It has the mouth opening with this little arrowhead, and then the other appendages associated with it. And then in panel E, we can see a cricket embryo, which also shows a labrum, which is much bigger and flap-like, also associated with the mouth opening, and then the rest of the appendages with them as well. So because of all of this combination of features, both the topology, the relationship with the mouth, and the morphology of the feature itself, I'm quite confident that this represents a labrum rather than something else. Now, whether this is a labrum is actually extremely important for interpreting the morphology or the segmental origin of these great appendages, because the labrum itself has very strong segmental implications for the head. So if we see this diagram, this is a very schematic germ band representation. So what we have in panel A is an early germ band stage, and the labrum originates at the very front of the germ band in association with the mouth in the protocerebrum. But then as all of the morphogenetic changes occur during embryogenesis and particularly organogenesis, the mouth and the labrum basically move backwards in the germ band until they basically end up sandwiched in between the first pair of limbs, or the pitucerebral pair of limbs. Because of this, if we see the labrum in an arthropod, we have a very strong sense of two things. One is that the protocerebral appendages are expressed as the labrum itself, and second one is that the first pair of appendages has to be due to cerebral, because the previous ancestral pair of appendages is already expressed in the labrum itself. Now, this carries the implication that the great appendages of megachirons are deuterocerebral. And this, of course, collapses the possible hypotheses because others have been suggested. So this is pointing towards a deuterocerebral affinity. But then more importantly, this actually concurs really nicely with recent discoveries of paleoneurological data in Cambrian megachirons, not only from the Xinjiang, but also more recently from localities in Utah from the mid-Cambrian, that show that there is clear consistency in the neurological preservation of these structures. So what we're being able to do is to basically tie this with a little pretty bow and say all of this data is actually resonating with each other rather than being in conflict. So the main implication here is that we can follow the evolution of the protocerebral appendages from being this ancestral frontal appendage as we see in onychophorans and radiodons, transforming into this hypostome labrum complex that we see in things like trilobites. And in the case of megachirons specifically, because the labrum is reduced quite significantly, this strongly suggests affinities with chelicerates, because chelicerates, unlike mandibulates, have a labrum, but the labrum is almost invisible in the adult form. So to conclude here, what we have been able to show is that micro-CT reveals very fine morphological details that basically revives the study of Xinjiang. I mean, not that they needed to be revived, but it really opens a new dimension to their study. The head organization of the encoelia has a reduced labrum associated with a posterior-facing mouth, which are important landmarks for understanding the segmentation of this animal. And the labrum clarifies the deuterocerebral origin of the great appendages, supporting likely affinities with the chelicerate stem lineage. With all of that, I just want to thank you for your attention. I would like to thank everyone in the lab at Harvard and welcome any questions that you may have.
Thank you very much, Javier. That was a fantastic talk. We do have some questions coming in for you. First one from Richie Howard. Javier, are we confident that the deuterocerebrum is homologous between onychophorans and arthropods? Is there any possibility that they are independent developments from a shared ancestral one-segmented brain?
Yeah, absolutely. So there is evidence that in the early evolution of onychophorans, basically onychophorans and arthropods, they developed the deuterocerebrum integration completely independently from each other. And this comes not only from development, but also from reconstructing the fossil record based on Lobopodians. So yeah, interesting convergences all the way around.
Great. I think we've got time for one more quick question. One from Emma Long. Great talk, Javier. Awesome models. Did you manually segment the CT data to generate the models? It would be cool to separate out the different structures, e.g. the labrum, and view them independently of the other structures.
Of course. So for some of this work, we do segment them out manually for highlighting particular structures. The models I showed are done with volume thresholding because it allows us to, in a single swoop, identify the pixels that are corresponding to the pyrite. And this gives us a slightly cleaner model that we have to tinker less with. But it is still necessary to do a bit of cleaning, like post-production, to get rid of all the particles because there will be pyrite in the matrix as well.
Thanks so much, Javier. There's one more question from Luke Parry for you in the chat. If maybe you could answer that during the next talk or something. But thank you so much. That was a really fantastic talk.